In a previous lesson, you learned about the form of cellular division called mitosis, and we talked about the stages of the cell cycle and mitosis. So as a quick review, let's go over those stages of mitosis again. So the cell cycle, which includes mitosis, starts with something called interphase. And recall that interphase is actually broken up into three kind of sub-phases. One of those phases is the G1 phase. The other kind of middle part of interphase is called the S phase. And then the third part is the G2 phase. And remember that the G1 and the G2 phases are primarily associated with growth and basically drawing together resources, preparing the cell for the next step in the cell cycle. Now the S phase, remember that something really important happens during the S phase, and that's where DNA replication occurs. Now, after interphase, the cell may go through the mitotic phase, which is when the cell would actually divide in order to make new cells. And the subphases of the mitotic phase can be abbreviated or remembered as PP, MAT, and then we can go ahead and put a C in there as well for cytokinesis. So remember that during the mitotic phase, we have prophase, prometaphase, metaphase, anaphase, and telophase, which usually happens kind of simultaneously with cytokinesis. If you need help remembering what happens during each of these phases, you should go back and watch the video about mitosis. But generally speaking, remember that what happens is that the chromosomes basically get split in two so that each new daughter cell gets a complete set of DNA. Now, as we mentioned, meiosis is another form of cell division, but as you learned in the introductory video, meiosis only happens in stem cells in the gonads with the goal of producing these specialized sex cells known as gametes, and the gametes include the eggs and sperm. So meiosis has a lot of similarities with mitosis, but it also has some differences. The first thing is that it starts out the same. So the first stage of meiosis is also called interphase. And just like with mitosis, meiosis's interphase also has a G1, an S phase, and a G2 phase. And what happens during these phases are identical to what is happening during mitosis. So go back and review. Remember that the G phases are primarily associated with growth. And the S phase is where we have DNA replication. And remember that S actually stands for synthesis, which is the same thing as saying that we're making DNA. So we're replicating DNA, we're making DNA. That's what's happening during that S phase. So interphase between mitosis and meiosis are identical. So these are the exact same between these two forms of cell division. Now, things start to go a little differently once we get past interphase, but there are still a lot of similarities between mitosis and meiosis. So the next phase is going to be something called Meiosis I. And we kind of designate the one with the Roman numeral 1, which could look like the letter I, or I typically just write it as a single line. So Meiosis I. Now, Meiosis I also goes through the phases of PP, MAT, C. Now you'll notice that we have a Meiosis I, which is probably giving you a preview that we're going to have a Meiosis II. So the stages of Meiosis I also get the Roman numeral 1 after them. So the first part of Meiosis I is going to be something called prophase I. Then we have prometaphase I, followed by metaphase I, followed by anaphase I, followed by telophase I, and a round of cytokinesis. So after Meiosis I is done, there is another round of growth that occurs, but we don't call it interphase 2 because it doesn't go through everything that the cell goes through during interphase. So the third step is actually something called interkinesis. So the key thing with interkinesis is that it is a period of growth. And so the cell is gathering resources, it's preparing to divide again, but the key thing is that there's no S phase. So you might want to give yourself a little star reminder next to interkinesis that DNA replication does not occur during interkinesis. Right? So no DNA replication. And this is really important. So make sure that you kind of write this down, put a star next to it, maybe highlight it, that there's no S phase, so no DNA replication happening during interkinesis. And we'll talk about why that's significant when we get into the nitty-gritty details of what happens during meiosis. Now, after interkinesis, we have meiosis II. Once again, the 2 is represented by the Roman numeral 2, which can either be just 2 lines like this, or you could draw it more like a Roman numeral 2 here. And what happens during meiosis II is another round of that PP mat C. So we have prophase II, prometaphase II, metaphase II, anaphase II, and I ran out of room, but after anaphase II, we would have a telophase II plus cytokinesis. Now, I want to point out a couple of more kind of similarities and differences between mitosis and meiosis before we get into the details of what happens during meiosis. So one thing I want to point out is the number of times the cell goes through the S phase in mitosis and meiosis. So remember, the S phase is when the DNA is replicated. So in mitosis, the DNA is replicated only once, and that's the same for meiosis. So in both meiosis and mitosis, the DNA is replicated only once. Now, both mitosis and meiosis have to do with cell division. So it has to do with a cell dividing into 2 cells in order to make more cells. Well, in mitosis, the cell goes through one round of cell division, and that happens during this mitotic phase. So this is one round of cell division. But in meiosis, we have 2 rounds of cell division. So we've got the meiosis I, in which the cell is going to divide into 2. And then we have meiosis II, in which the cells that were created during meiosis I are going to divide into 2 cells again. Let's do a quick review of what happens during these phases that we introduced in the previous slide, and kind of talk about how they are maybe similar or different to mitosis. So what happens during interphase in meiosis is exactly the same as what happens during interphase of mitosis. So first you have the G1 phase, And the G1 phase, remember the cell is preparing for the S phase. So it's preparing for DNA replication. And so the cell is growing and it's collecting things like nucleotides, it's making protein so that it has the enzymes needed for DNA replication, etc., etc. After the G1 phase, we have the S phase. Remember, S stands for synthesis. And what is being synthesized, remember synthesis means to make something. So what's being synthesized is another copy of DNA. So DNA gets replicated so that each chromosome gets copied. And so now after the S phase, each chromosome is made up of two connected sister chromatids. So remember that before the S phase, the chromosomes look like these bar-shaped chromosomes. After the S phase, the chromosomes look like X's. And you can see that on the picture up here where we have before the S phase happening up here. So this would be like during the G1 phase. The chromosomes would look like these bar shapes. This arrow right here stands for the S phase. And so remember, that is our DNA replication. And then we can see that the next kind of circle is showing us the G2 phase, which would be after the S phase. After DNA replication occurs, the cell goes through another growth phase. So that would be the G2 phase. And so the G2 phase is just another kind of round of growth where the cell is preparing to divide. And so for cell division, the cell needs to get energy reserves, it needs to make proteins, it needs to make enzymes. It's just getting all of those resources needed for it to go through a round of cell division. Now, it's really important that you understand the difference between homologous chromosomes and sister chromatids because during meiosis, different things are getting separated at different rounds of cell division. So you need to know what they are so that you know what's happening as they start to get separated and moved around during these cell division events. So as a reminder, homologous chromosomes are paired chromosomes. So whenever you see the term homologous, you should immediately think of the word paired. So homologous chromosomes are these paired chromosomes in which you get one chromosome from your mom and one chromosome from your dad. Or really, you get 23 chromosomes from your mom and 23 from your dad, but you get two copies of each chromosome, one from your mom, one from your dad. Another term that goes along with the word homologous and paired is the word diploid. So you should essentially think of these three terms. They're not the same thing, so you can't use them interchangeably, but they go together. So if there are homologous chromosomes present, it means that there are paired chromosomes present, and it means that the cell is diploid. So those three terms go hand in hand. And the reason why homologous chromosomes exist is because of sex. So remember that you get one set of chromosomes from your mom that's kind of packaged in this egg, and then you get one set of chromosomes from your dad that's packaged in a sperm. Now, homologous chromosomes might look like just two bars, or it might look like two X's. So it really just depends whether you're looking at the homologous chromosomes prior to the S phase or after the S phase. So if we were to kind of draw this up here, let's say you've got mom in red, and we'll do dad in blue. So let's say you get these 23 chromosomes from your mom, and then you get your 23 chromosomes from your dad. Now, before the S phase, these chromosomes are just going to look like these linear bar-shaped chromosomes. But after the S phase, remember that these chromosomes just get another sister chromatid attached to them. So after the S phase, they might look like an X. Before the S phase, they just look like a line. So it doesn't matter whether it's a single line or these X-shaped chromosomes, as long as you have a set that you got from mom and a set you got from dad, they're still considered homologous chromosomes. A couple other things about homologous chromosomes that are important to note is that they are not identical to each other. They are similar. They carry similar genes and similar types of information, but you might get different versions of it. So you can think of chromosomes as carrying these recipes for how to make proteins in your body. And we're going to talk about that more when we talk about genetics and protein synthesis. But you can think of it as, let's say, homologous chromosome pair number 1, let's say it carries the genetic information on how to determine how tall you're going to be. Now, both of them include recipes on height, but let's say that your mom gives you genetic information that says that you're going to be shorter, and your dad gives you genetic information that says you're going to be taller. It's kind of like if you were to get 2 copies of recipes for cupcakes, but one copy of the recipe was for vanilla cupcakes and the other copy of the recipe was for chocolate cupcakes. So there's still recipes on how to make cupcakes, they're just slightly different variations or versions of those recipes. Now, if we take a look at sister chromatids, so sister chromatids are identical. So this is important to note. They are identical to each other because they are formed during the S phase. So formed during the S phase. Remember that during the S phase, we have DNA replication. And so what's happening is that a chromosome is making a copy of itself and then attaching that copy to itself. So it would be like if you were to take a piece of paper, take it to a copy machine, make a copy of it, and then staple the 2 copies together. So you don't have any new information. You have all of the exact information that you started with. You've just made another copy of that information. So let's do a little recap about the difference between homologous chromosomes and sister chromatids. So homologous chromosomes, remember, are paired chromosomes. And if they exist, a cell is diploid. So the terms homologous, paired, and diploid go hand in hand. So if you have homologous chromosomes, it means you have paired chromosomes, and it means that the cell is diploid. Now, the reason why these homologous chromosomes exist is because of sexual reproduction. So they exist because of sex. And that's because you have an egg that gets fertilized by a sperm, and the egg contains one set of chromosomes. The sperm contains a set of chromosomes, and when they combine to form the zygote now has these paired chromosomes in which it got one set of chromosomes from the mom and one set of chromosomes from the dad. Now, homologous chromosomes are not identical to each other. So they're not identical, but they are similar in that they carry the same type of genes, but they may contain slightly different variations of the same gene. So we can use the example of eye color, where you might end up with a chromosome from your mom and a chromosome from your dad, and they both code for eye color. So they both contain the recipe for how to make the color of your eyes. But the different variations might be that your mom gives you information on how to make blue eyes, and your dad gives you information on how to make brown eyes. Now, homologous chromosomes can look different depending on whether you're looking at them before the S phase or after the S phase of interphase. So remember that before the S phase, the chromosomes are going to be these bar-shaped chromosomes that are paired up with each other. After the S phase, each one of those chromosomes is going to look like an X, but the important thing is that they're still paired up with each other. So in both of these cases that we see down here at the bottom, we're looking at homologous chromosomes. We're just looking at the homologous chromosomes before DNA replication has occurred and then after DNA replication has occurred. Now, when it comes to sister chromatids, they may exist in haploid cells or they may exist in diploid cells. It just kind of depends on when you are looking at the presence of these sister chromatids. So you can't necessarily say that sister chromatids are always associated with a haploid cell or always associated with a diploid cell because it might be present in either condition. The reason why sister chromatids exist is because of the S phase. So they exist because of DNA replication. Sister chromatids are identical to each other. So they are literally exact copies of themselves. Chromosomes that contain sister chromatids always look like X-shaped chromosomes. So these X-shaped chromosomes are showing us that there are two sister chromatids that are attached to each other. If you see a bar-shaped chromosome like this, it means that the S phase has not occurred, and it means that sister chromatids do not exist yet. So if you see X-shaped chromosomes, it means that you have sister chromatids. And so the example that we can use for sister chromatids is like taking a recipe, putting it in a copier, making an exact copy, and stapling those two copies together. So let's take a look at some possible combinations of chromosomes and cells to kind of help you understand the difference between homologous chromosomes and sister chromatids. So you may want to pause this video and see if you can figure out which ones of these cells have homologous chromosomes present and which ones have sister chromatids present. And remember that it's possible for a cell to have both homologous chromosomes and sister chromatids present at the same time. So those two terms are not mutually exclusive. All right, so hopefully you pause the video and try to figure this out on your own. So let's go over the answers. So in this first cell here, are homologous chromosomes present? And the answer is yes. And we know that because these chromosomes are paired up. So remember that paired chromosomes equal homologous chromosomes. Are sister chromatids present? And the answer to that one is no. Remember that sister chromatids are only found in the X-shaped chromosomes, and since these are bar-shaped chromosomes, we do not have sister chromatids present. In our second cell, we do still have homologous chromosomes because we still have those paired chromosomes. So the answer to that one is yes, we have homologous chromosomes. We also have sister chromatids. Because we have those X-shaped chromosomes present and those two kind of arms of the X, each one of those arms equals a sister chromatid. In our last example, we no longer have homologous chromosomes, and that's because we don't have those paired chromosomes anymore. However, we still have sister chromatids, and that's because we have those X-shaped chromosomes present. Now, going into the event of Meiosis I, we can first look at prophase I. And some of the events of prophase I are very similar to mitosis, but there are some differences. So first of all, just like in mitosis, the nuclear envelope breaks down. And that's because, remember, these chromosomes need to be able to move around into the new daughter cells. So we have to get rid of that barrier. We have to get rid of that nuclear envelope. Now, one thing that happens that is very different from what happens in mitosis is this thing called synapsis. And what happens is that these homologous chromosomes pair up. And you can kind of see that in these pictures over here. You can see that these are homologous chromosomes. And you can see that they are pairing up with each other. So they're hanging out next to each other. Now, this did not happen in mitosis. Now, the significance of this synapsis, so the significance of these homologous chromosomes pairing up, is that something called crossing over, which is also known as genetic recombination, occurs. And you can see that happening here, where basically what happens is that one of the arms, or part of an arm of one homologous chromosome, is going to get exchanged with an arm or part of an arm of the other homologous chromosome. So you can see in this picture down here at the bottom that our sister chromatids are now no longer identical to each other. So I mentioned before that we said sister chromatids are identical, and that's true at first until they become not identical. And so this genetic recombination is actually really important because it helps to increase genetic diversity. And we won't get into a lot of the details about evolution and natural selection, but generally speaking, increased genetic diversity means a better or healthier population of humans. So as you increase genetic diversity, you increase the health of the population. Now, this increase in genetic diversity, it's not going to affect you, but it could affect your offspring, or it could affect the next generation. Just like when this happened in your parents, as they were making gametes, it didn't affect their health, but it could have affected your health and the health of the human population in the next generation. During prometaphase 1 of meiosis I, the nuclear membrane has totally dissipated at this point, and the mitotic spindle is starting to develop. So even though we're talking about meiosis, it's the same kind of structure of the mitotic spindle, which remember is equal to centrioles plus microtubules. And the mitotic spindle is what is going to ultimately move these chromosomes around and separate them into the new daughter cells. So on these chromosomes, there are these little connection points called kinetochores. And these kinetochores create kind of like a binding spot for these microtubules of the mitotic spindle to attach to. And so that's ultimately what allows the mitotic spindle to pull the chromosomes around, and then ultimately at some point it's actually going to pull the sister chromatids of the chromosomes apart. During metaphase 1 of meiosis I, what happens is that the homologous chromosomes line up at the center of the cell. So remember that these homologous chromosomes paired up during synapsis during prophase. So essentially what we would have is this cell that looks kind of like this, and you would have these homologous chromosomes that are paired up and lined up along the middle of the cell. Now, kind of one interesting thing is that which side each homologous chromosome is on is random, and this is actually called independent assortment. So in this drawing I drew up here, this is option 1. Okay, so this is like one option for how these chromosomes could line up during metaphase 1. But they could have just as easily, we'll say this is option number 2, we could have ended up with a situation where the chromosomes line up like this. Or we could have option 3, in which the chromosomes line up like this. And the reason why this matters is because what happens during anaphase is that these homologous chromosomes are going to get separated from each other. And ultimately, one of those homologous chromosomes is going to go to one side of the cell, the other one's going to go to the other side. And you can see that depending on how these line up, you're going to get a different kind of arrangement of these chromosomes in the daughter cells. And you'll notice here that I'm only drawing 3 sets of chromosomes, but remember that humans have 23 sets or 23 pairs of chromosomes. And so you can imagine how many different possibilities there are with regards to what side of the cell the chromosome from mom lines up compared to the chromosome from dad. So you end up with these almost infinite possibilities of how these chromosomes can line up and separate into the daughter cells in different ways. So this kind of randomization of how these homologous chromosomes paired up is called independent assortment. And you maybe have guessed that the purpose of this is to increase genetic diversity. So remember that if we increase genetic diversity, it can make the population of humans healthier overall. This image is just showing you different possibilities of how the chromosomes can line up during metaphase 1. So this is during, remember, metaphase 1, in which the homologous chromosomes are pairing up with each other and then lining up kind of in the middle of the cell. So you can see in this top option here, so this would be like option number 1, and then down here we would have option number 2. You can see in option number 1 that we end up with the blue chromosomes on the left and the red chromosomes on the right. So when we go through the first round of meiosis, or the first round of cell division, these are what the daughter cells end up looking like. Now down here for option 2, you can see that the top pair of chromosomes, you've got the blue one on the left and the red one on the right, and the bottom pair, you've got the red one on the left and the blue one on the right. And so what happens is we end up with this other possible kind of situation occurring in the daughter cells. Now we haven't even talked about the kind of the next round of cell division yet, so we'll get to that in a second. But you can see here that whatever happens during independent assortment is ultimately going to affect the end results of which chromosomes end up in which gametes. During anaphase 1, the homologous chromosomes that lined up in the middle of the cell during metaphase 1 get separated from each other. And this separation is called segregation. The site that each chromosome goes to during segregation is random due to that independent assortment that we talked about, which happens during metaphase 1. And remember that the independent assortment just has to do with which side the homologous chromosomes are lining up when they line up during metaphase 1. Recall that the homologous chromosomes are not identical, because remember, you're getting a set from your mom and you're getting a set from your dad. And these homologous chromosomes are these paired chromosomes. Now remember that the chromosomes have similar recipes on them, but potentially different variations of those recipes. So just like we talked about cupcakes, you essentially inherit 2 recipes for how to make cupcakes, but one recipe might be for chocolate cupcakes and one recipe might be for vanilla cupcakes. Just like with, a more realistic example would be something like eye color. So it could be that your mom gives you a recipe for blue eyes, whereas your dad gives you a recipe for brown eyes. So we mentioned how during metaphase 1, the order that these chromosomes line up might differ. So remember, we've got these examples where the chromosomes from mom might all line up on one side of the cell, and the chromosomes from dad might line up all on the second or on the other side of the cell. Or we could get kind of a variation in which some of mom's chromosomes line up on the left and some on the right, and some of dad's chromosomes end up on the left and some end up on the right. And the reason why this matters is because during anaphase 1, remember that these homologous chromosomes are going to get separated. And you're going to have some of those homologous chromosomes going into one of the daughter cells and some going into the other daughter cell. So, depending on how these are lined up, we end up with a kind of a unique combination of these genes that end up in your gametes. So essentially, what happens is that you inherit these genes. So you inherit these recipes from your parents. And then when it's time for you to make gametes, what happens in your cells is that these recipes or these genes get kind of shuffled. So it's almost like taking a deck of cards and shuffling them and then dealing them out in a new way. And so essentially, your gametes, you're just taking the genetic information you inherited from your parents and you are shuffling them so that each of your gametes ends up with a unique combination of genetic information, from some of it coming from your mom and some coming from your dad. And telophase 1, similar to what happens during telophase and mitosis, those separated chromosomes arrive at opposite sides of the cell. And then cytokinesis happens, in which the cell membrane starts to pinch in, and that cell membrane ends up dividing the cell into two new daughter cells. So let's summarize what we have at the end of meiosis I. So remember that we started with this cell kind of going through the G1 phase, and then it went through the S phase of interphase, and then it looked like this during the G2 phase. Now, this first set of arrows is representing what happens during meiosis I. So at the end of meiosis I, we have two daughter cells, and those two daughter cells contain 23 total chromosomes. But each one of those chromosomes is made up of two chromatids. So each one of these cells is going to have 23 total chromosomes and 46 total chromatids. So let's take a look at what's happening with regards to our homologous chromosomes. So at this stage in meiosis, do homologous chromosomes still exist? So do we still have paired chromosomes? And the answer is no, because during meiosis I, the homologous chromosomes get separated. So that's kind of the key thing that's happening during meiosis I, is the homologous chromosomes separate during meiosis I. So if we no longer have homologous chromosomes, are the daughter cells haploid or diploid? Hopefully you answered haploid. And that's because remember that the term diploid goes along with the term homologous. So a cell is only diploid if it has homologous chromosomes, meaning that it's only diploid when paired chromosomes are present, because paired chromosomes are the homologous chromosomes. So since we separated the homologous chromosomes during meiosis I, it means that we no longer have homologous paired chromosomes, which means that the cell is no longer diploid, so now the daughter cells are haploid. So if we go over to this picture over here, this first cell that is kind of starting out this process is going to be diploid, and that's because we have homologous chromosomes. So we have homologous paired chromosomes. After the cell goes through the S phase of interphase, the cell is still considered diploid because we still have those paired homologous chromosomes. But because during meiosis I, the homologous chromosomes separate, these cells are now known as haploid. So these cells no longer have these paired homologous chromosomes. Now the chromosomes are still X-shaped because we still have sister chromatids, but we do not have homologous chromosomes.